The goal of this experiment is to determine factors that control the release of a drug from a hydrogel bead. The drug is simulated by the dye tartrazine, commonly used as a food coloring. The hydrogel bead represents the capsule. Our goal is to determine which conditions lead to the greatest fraction of dye being released. The beads made in this experiment to simulate a drug being administered to the body are made from alginate, calcium ions, and tartrazine. Alginate is a polymer commonly used in drug delivery studies due to its biocompatibility. The process of making the beads is as followed. The sodium alginate is exposed to calcium ions, and certain monomers form in the polymer, causing it to fold around the calcium ion, allowing the hydrogel bead to form. This is called cross-linking. Before the cross-linking occurs, the drug can be mixed with the alginate so that it gets suspended in the hydrogel bead, where it can later be dispersed into the body, or in this case, our water bath. Several factors contribute to the rate of drug being released. For example, the size of the bead, the temperature of the water bath, the alginate concentration, the mixing speed, and the cross-linking time. The bead sizes chosen were based on the availability of syringes in the lab and are small, medium, and large. The mixing speeds chosen were 200, 300, and 400 revolutions per minute, and the cross-linking time was 1, 3, and 5 minutes. When these variables were not being changed, the following were being held constant. The medium syringe size, or 19G times 1.5, 300 revolutions per minute, and 3 minutes. The governing equation for our experiment is that of the fraction of dye being released, where F is the fraction of dye being released, K is a constant that incorporates characteristics of the macromolecular network and the drug, and N is the diffusional exponent. F can be calculated by dividing mt over m infinity, where mt is the mass of dye released at time t, and m infinity is the mass of dye released at equilibrium, or our final data point. For pure Fickian diffusion of a sphere, n should be equal to 0.43. Taking the natural log of equation 1 yields equation 2, which helps us find k and n. N is the slope of the line, and k is equal to the natural log of k. In order to determine the concentration of dye released at each time point, a calibration curve was made using known dilutions of tartrazine. Each of these dilutions were made using a UV-Vis spectrophotometer so that a relationship between absorbance and concentration could be made. These measurements resulted in the following calibration curve.
The first solution that needs to be made is a stock solution of tartrazine, which is done by measuring 0.5 grams of tartrazine for every 1 liter of DI water. The DI water was measured using a graduating cylinder and poured into a 1 liter container with a stir bar. The tartrazine was added to the water and stirred until all clumps of the dye dissolved. It is important to make sure to cap the container to ensure that the DI water does not evaporate while the solution mixes. The next solution that needs to be made is the alginate solution. To make 300 milliliters of this solution, 3 grams of alginate is added to 300 milliliters of the tartrazine stock solution. The solution is left to mix on top of a stir plate with a stir bar until all the alginate dissolves and there are no more clumps in the solution. To make 6% by weight calcium chloride solution, 500 milliliters of the tartrazine stock solution was measured, and 300 grams of calcium chloride was also measured out and added to the stock solution. The mixture was then left on the stir plate with a stir bar until the solution became a homogeneous mixture. Once all of the solutions are made, the beads are ready to be made. To begin, pour the calcium chloride solution into a weight boat and then set aside. Afterwards, load a syringe with 3 milliliters of the alginate solution. Then carefully twist the needle tip onto the syringe and begin to dispense the alginate into the calcium chloride droplet by droplet. Try to avoid drops falling on top of each other by moving the needle around because the beads may start to stick to each other and create malformed beads, or the beads may combine to make a bigger size. The beads are separated by pouring the contents of the weigh boat into a funnel lined with filter paper. The beads were then rinsed with DI water using a clean syringe filled with the water. After rinsing, the beads were placed into a 150 milliliter beaker filled with 100 milliliters of water. Once placed in the beaker, the solution was stirred for an hour. Absorbance of the solution was taken every 2 minutes for 10 minutes, every 5 minutes for 20 minutes, and then every 10 minutes for 30 minutes. The absorption of the solution was taken by pipetting the solution into a clean cuvette, filling the cuvette at least 3/4 of the way, and then using a UV-vis spectrophotometer to take these measurements. 3 tests were done for each of the variable changes, and the data was recorded. The solution of the cuvette was then returned into the beaker that it was taken from. These measurement steps were repeated again until all of the tests were done.
For the data collection, 3 variables were changed and measured in triplicate. For this experiment, the changed variables were bead size, RPMs, and cross-linking times. Once all the data was collected, the averages for each trial of each variable were plotted. The bead sizes used were 16, 19, and 22, which were the needle sizes provided. Cross-linking time and RPMs were kept constant at 3 minutes and 300 RPMs, respectively. The RPMs used were 200, 300, and 400 RPMs, while cross-linking time and bead size were kept constant at 3 minutes and size 19. The cross-linking times used were 1, 3, and 5 minutes. Bead size and RPMs were kept constant at size 19 and 300 RPMs. From these data plots, which measure absorbance versus time, the concentrations were found at each absorbance using the calibration curve. And once the concentrations were found, the fraction of dye released, F, was found by dividing each concentration by the final data point at 60 minutes. These were then plotted versus time for each variable. Then taking the ln of these graphs can determine the n and k values by taking the line of best fit and using the slope as n and the y-intercept as the ln of k. Here are all the n and k values found tabulated. As seen, the values of n decreased when increasing RPMs and the k values increased, and the n values increased when cross-linking time and bead size was increased and the k values decreased. Increasing the bead size increases the n values, making the beads closer to the 0.43 value for n for a spherical shape to allow better diffusion, but the k values decrease more abruptly, so the transfer rate is more optimal at the smaller bead size 22, which is likely due to the smaller surface area at which the diffusion must occur. Increasing the RPMs increases the transfer rate. However, higher RPMs create vortexes, which promote bad mixing and may not distribute a drug efficiently throughout the body. Therefore, of the RPMs tested, the optimal RPM would be 300. Finally, for cross-linking time, 3 minutes was the optimal time. This is because cross-linking for 1 minute would be inefficient for delivering the drug into the body because it may diffuse too quickly and improperly distribute the drug throughout the body, while 5 minutes could be too long to allow for cross-linking because it creates stronger bonds among the polymers that may further inhibit diffusion.